Afternoon. Hi, my name is Art Arnold. I'm director of the Laboratory of Neuroendocrinology at UCLA for another few days. I'm stepping down at the end of this month, and Paul Misavage is going to be director starting next month. Uh, it's I always say every year, some of the things I say every year, but I think they're worth repeating. This is the high point of the neuroendocrine calendar at UCLA. It's the Charles Sawyer Distinguished Lecture. And we remember Tom Sawyer today, who was a great leader at UCLA and internationally in the field of neuroendocrinology. Tom really established neuroendocrinology at UCLA and built a group around himself and really brought together a federation of laboratories, which eventually became the Laboratory of Neuroendocrinology of the Brain Research Institute. This is always this really serious picture of Tom, but he actually was a funny guy. This is a picture of him with 2D glasses with his students looking at, I think, some anatomy diagrams. Here's a picture of him with his big smile. So Tom was a gentleman and a scholar. He's was chair of the Department of Anatomy, which is now neurobiology, and he taught gross anatomy for many years, and was. This is actually a picture. It says an appreciation to his many years of excellence in teaching and concern for students. It was an award that he got in 1981. And his work was really seminal in the development of the understanding of the relationship between the brain, starting in the late 1940s and through into the 1980s, the relationship between the brain and the gonads, and the interrelationship and how the brain controls the gonads and vice versa. So the two Nobel Prize winners who discovered releasing factors in the hypothalamus that control the pituitary, Yeaman and Shalley, both had very laudatory things to say about Tom, which are here. Yeaman said that he asked the right question or provided a clue to the right answer. His many students and early collaborators are now some of the leaders of the expanding field of neuroendocrinology. So I've been at UCLA for a long time, but once I came here, I found that if I would go to an international meeting, always somebody would come up to me and say, I used to be at UCLA when I was a postdoc, and I trained with Tom Sawyer, because Tom had a grant that brought in international scholars. And so he really developed this international community. Some of his great discoveries were in the late 40s. It was published in 1950. He showed that the release of LH causing ovulation was linked to a 24-hour periodicity, because you could inject barbiturate and delay the release of LH and therefore ovulation, but you delayed it by a full 24 hours. You didn't delay it for so the timing was linked to the circadian rhythm. The famous paper by Kawakami and Sawyer in 1959 was some of the first evidence of direct effects of steroids on the brain, measured electrophysiologically by the EEG, and he had profound effects on the understanding of puberty. So this is a list of members of the UCLA Lab of Neuroendocrinology in the current iteration of the training grant, which was just submitted by Paul Misavage. The training grant's has been funded for 35 years, and of course we'd like to continue that. So the Sawyer Lectureship has been every year since 1998. This is a list that any neuroendocrinologist would like to be on. It's a list of highly distinguished people. And this year we're really pleased to welcome back to UCLA Martin Kelly from the Oregon Health and Sciences University. When I was a student, there were rumors that estrogens could have rapid effects on the brain. And this was a huge puzzle, because the evolving understanding of the receptors for estrogens placed those receptors in the nucleus, and the estrogens bound to these nuclear receptors and change the DNA, which changed the readout of genes. And this evolving understanding in the first maybe 20 years of my career was a slow effect of estrogen, okay? But there were a few people who discovered rapid effects early on, and among those were Martin Kelly, and he's really made huge contributions to our understanding of these rapid effects, which are membrane effects, and they're membrane estrogen receptors. So the Sawyer Lectureship is the person who's invited every year is invited by vote of the trainees of the Laboratory of Neuroendocrinology, and it's they who choose the distinguished lecturer based on the accomplishments of the lecture. So the other major problem I think Martin has, besides rapid membrane effects, Martin's as you'll see immediately, he's an electrophysiologist, and there aren't very many electrophysiologists left studying hypothalamic processes at this using cell biological methods, and so he has a special niche there. But the other major issue is that people have known for a long time that the same neurons in the hypothalamus affect totally different, what we think of in our own minds and our own models of the brain, totally different physiological functions. For example, regulation of food intake and reproduction, okay? The same neurons are involved in these, and so these neurons and areas of the brain and molecules are integrating signals that are relevant to both of these systems because of the need to coordinate metabolism and reproduction. So Martin's talk today is entitled Integration of the Anorexigenic Effects of Estradiol and Metabolic Hormones by Hypothalamic Arcuate Neurons. So welcome back, Martin.
Okay, I want to start by thanking Art and the students of neuroendocrinology for inviting me to give this lecture, and I'm deeply honored to be one of the chosen few, if you will, to talk about Tom Sawyer. And so I'm just going to say just a few brief words, and I'll talk more about my history, but I really consider myself a scientific godson of Tom Sawyer, and he was really, I've known his works from the 50s when I was an undergraduate student at the University of Michigan doing an honors project, and actually it's ironic that Art put up that paper by Everett and Sawyer showing that barbiturate blocked the LH surge, and we actually repeated that, used that model as and this is an undergraduate study, and really led me into my quest to really look at the rapid effects of estrogen on hypothalamic neurons. And I should say briefly that Tom Sawyer was responsible for my first job at University of Pittsburgh because he came up to me in 1997 at the International Physiology meetings in Paris and said that, you know, Ernie Nobile's going to contact you about a job that he called me about, and he recommended me for this job. And you know, it's really, I have Tom to thank for that first position to look at. Today I want to really cover three topics, and hopefully I have enough time to get through all these, but I want to start out just briefly talking about 17-beta estradiol signaling and contrasting it in the anorexigenic POMC neurons and the orexigenic NPY-AGRP neurons in the medial basal hypothalamus. So this is the yin and yang in terms of controlling energy homeostasis. And then I want to then talk about some more recent findings in terms of the convergence of insulin and leptin signaling to activate a certain class of channels in POMC neurons that leads to the excitation of POMC neurons. So like estrogen, leptin and insulin are anorexigenic, and therefore they converge in POMC neurons to ultimately regulate energy homeostasis. And then at the end, touch on some very new findings in terms of talking about how does estrogen protect insulin signaling with diet-induced obesity.
So is your microphone on?
Can you hear me in the back?
Yep.
Okay. So Going back to my postdoc in Germany at the Max Planck Institute, I started using the guinea pig as a reproductive model because it has a long reproductive cycle, more like the primate. It's 18 days, and there's a true follicular and luteal phase that we could divide up or look at an estrogen-driven phase versus a progestin-driven phase, just like in the primate, versus the rat and the mouse, where everything is a gamish in a sense. So this is just showing this model, and what you can see here is that in the late follicular phase in which you have vaginal openings, so the animals now are becoming reproductively active, you can actually see a decrease in food intake during this phase. And this is what fascinated me, and when I moved back to the United States, I actually proposed to look at this integration between reproduction and energy homeostasis. And at the time, and this is going back to the early 80s when no one really cared about the hypothalamus, because the joke was this is like recording from the nucleus down under. I had proposed that the pro-opiomal anacortin neurons were really the command neurons of the hypothalamus, and they were involved in integrating all these homeostatic functions. So at the anatomical level, at the EM level, it was shown by Ann Silverman, Paul Goldsmith, noted electron microscopists, that the POMC neurons showed all these various synapses throughout the hypothalamus. And so the other thing that we noted is that these, and became a center of focus, was these pro-opiomal anacortin neurons. They express autoreceptors, and these are the mu-opiate receptors that are expressed in these pro-opiomal anacortin neurons, or I'll also call them the POMC neurons for short. And they're highly endowed with these mu-opiate receptors. And this is just an in situ hybridization from Alina Renniclave's lab, and it's just showing the silver grains over these immunostaining of beta endorphin. And this is highly localized. This is the mu-opiate receptor down in the medial basal hypothalamus. So basically all the neurons in the arcuate express this receptor, but we were particularly interested in the mu-opiate receptor, or the mu-opiate receptor expression in the POMC neurons. And the mu-opiate receptor and the GABA-B receptor in these pro-opiomal anacortin neurons are coupled to the G protein coupled into rectifying potassium channels, such that when you activate the mu receptor or the GABA-B receptor, you have this membrane-eliminated event where the beta-gamma subunit shuttles over and activates these GERK channels that causes a membrane hyperpolarization. And so the early studies, and I'm just quickly taking you through the last century, if you can, and please interrupt me if you have any questions. Early on, I showed that the mu-opiate receptor very robustly hyperpolarized. So this is a POMC neuron firing along here, and just briefly applying this selective mu agonist DAMGO, it's an enkephalin analog, you can see this membrane hyperpolarization that inhibits the cell firing. And in 1992, I published that if you treat with estrogen over 24 hours, you would see a uncoupling or a desensitization of this mu-opiate response. And then we went on after 92 to really look at the rapid effects of estrogen to cause this, what we now know as a heterologous desensitization. And I'm just showing one example here. So now we have the cell in TTX to electrically isolate the cell, so we can say that these effects are directly on the neuron that we're recording. And this is just showing a current clamp recording where you see a dose response to this mu agonist, and you can see this membrane hyperpolarization. And then when one starts the co-perfused 20 nanomolar estrogen, you can see this reversal of this membrane hyperpolarization. That is the uncoupling of the mu receptor from activating the GERK channels. So this we now know is a heterologous desensitization, and we moved on. Those were sharp electrode recordings, and then we moved on to whole cell recordings in which we could do whole cell recording from the neurons and do a number of manipulations, including intracellularly perfusing different reagents to look at the signaling pathway, but also they'll allow us the opportunity to harvest the cell contents afterwards to measure the expression of different mRNAs. And this just shows the voltage clamp experiment where you see now in electrically isolated cells, you can see this outward current due to baclofen or damgo, and you can very readily wash this out. So our model, again, is initially was the overreactive mice guinea pig, where we're looking at these rapid, if you will, 17-beta estradiol effects. But we now have also incorporated mice because of the ability to use different transgenic mice. So again, this is summarizing a lot of work from the last century and bringing you into this century. What we elucidated was a GQ-coupled receptor that would activate phospholipase C, PKC delta, PKA, that would eventually uncouple these cells, that is, desensitize mu-opiate GABA B receptor. But any GI-GO coupled receptor in POMC neurons, this has, it hits all of these. And what we knew is that we could use estradiol conjugated to BSA, and we could still get these effects. So we limited the ability of 17-beta estradiol to penetrate the membrane, and we could still activate the signaling pathway. So what we had was a membrane estrogen receptor that was G-alpha-Q coupled to phospholipase C, PKC, and PKA to cause this desensitization. And I don't have time to go through all the data where we use all different inhibitors, activators of the different signaling pathways to elucidate how the signaling worked. But what became a major point of focus was the fact of what is the nature of this membrane estrogen receptor, and can we use a more selective agent to target this? And so over the years, and this is going back to my studies as an undergraduate even, 17-beta estradiol was a full agonist in this scenario. 17-alpha estradiol was without effect, and more recently, or in this century, if I can still use that term, tamoxifen was inactive. 4-hydroxytamoxifen was a partial agonist, as was raloxifene. And ICI, which we now know is fulvestrant, it's used in the clinic for breast cancer treatment. This was an antagonist at 0.3 nanomolar. So we know this was an estrogen receptor. It was highly stereoselective, and it was antagonized by known antagonists for the estrogen receptors. Now, this finding that 4-hydroxytamoxifen was a partial agonist really led to a collaboration with Tom Scanlon, who was a chemist at UCSF at the time. And we knew that tamoxifen existed in both an E and a Z conformation about this carbon-carbon double bond. And it was through Tom's chemistry that he came up with the idea of putting an amide linkage that would lock this into the E or the Z conformation. And it was lo and behold, what we found was the E conformation, what we called STX. It was an STX and STY, but it was the the E conformation that was more potent than estrogen in terms of activating this pathway, this desensitizing pathway. And this is just now on the y-axis here is the attenuation of the GABA B response. And I'll show you some traces how we measure this, but you can see in this dose response curve that STX is more potent than estrogen to cause this effect in POMC neurons. And we went on to show that these effects of estrogen, and that is 17-beta estradiol and STX, were fully efficacious in the ER alpha, ER beta knockout. These are the transcription factor This is a double estrogen receptor knockout mice. And you can see here that, in terms of attenuating either the GABA B response or the mu opiate response, you see this 40% attenuation that happens over the course of minutes using either 17-beta estradiol or STX, and both of these are blocked by the antagonist ICI. So what we had then was a ligand that would specifically activate this GQ membrane estrogen receptor that would cause this downstream signaling to increase the excitability of the pro-opioid melanocortin neurons, because it was removing a tonic inhibitory input onto these neurons. Now, several years ago, the question then came up, well, what is the action of in the NPY neurons? Because we know that in terms of this yin and yang signaling, that the orexigenic NPY neurons are also very critical. And this is just showing a micrograph showing from Todd Stinsick, postdoc in the lab, showing POMC neurons that have been labeled with a M-cherry channel rhodopsin, and this mouse is crossed with an NPY GFP expressing mice. And you can see this, the close interaction between the POMC neurons and the NPY neurons. So this is a coronal section through the medial basal hypothalamus, third ventricle, and the pituitary gland would sit down here, and this is the median eminence. And a very talented graduate student, Eric Smith, tackled this question in the NPY AGRP neurons that are existing down in the hypothalamus here. And this was really the first time where we could show in one cell type, one isolated cell type, both an ER alpha signaling mechanism that was distinct from this STX, or this GQ membrane estrogen receptor. So I'll take you first through that. So this is the sort of, now this is a GABA B agonist, that just like the mu opiate agonist, it's a voltage clamp experiment in whole cell. You apply your agonist, you get this outward current, and then you wash that out, you apply STX or 17-beta estradiol, et cetera, and then you redo this, and what you see is that STX is not diminishing that response, but is actually increasing it. And that increased response is again blocked by ICI. Whereas in contrast, 17-beta estradiol is acting just like it does in POMC neurons, where it's actually attenuating the response in the NPY AGRP neurons. And this is via a distinct signaling pathway. It's a PI3 kinase signaling pathway, and this is using a selective P110 beta inhibitor, TGX, that was produced by Keva and Chokat's lab at UCSF. And you can see that very effectively blocked these effects of 17-beta estradiol. But more importantly, using the Katzenell-Bogen compound that is selective for ER alpha, this is PPT, we can mimic these effects of 17-beta estradiol on the GABA B response. That is, it's diminishing or attenuating the response. So here we had a contrast between the GQ membrane estrogen receptor response and the ER alpha signaling response. And we also showed with the single cell RT-PCR that ER alpha transcript is expressed in these NPY AGRP neurons, but we know it's differentially coupled between these two neuron populations. So in the NPY AGRP neurons, with the orexigenic neurons, which drive food intake, that the STX is potentiating the effects of NPY, whereas in the POMC neurons, it's actually inhibiting the GABA B response and increasing the excitation of the POMC neurons. And this really explains some results that we had published a number of years ago, that STX was actually very effective in terms of its more effective than 17-beta estradiol in terms of inhibiting NPY expression. If we gave it systemically and actually looked at the MRA expression in the arcuate nucleus, that STX is more effective than estrogen in terms of inhibiting NPY expression. So the other question that then came up more recently is, how do these neurons communicate? So some years ago, there was shown a connection between the NPY-AGRP neurons that synapse on the POMC neurons, and via releasing GABA inhibits POMC neurons. So the question is how does the POMC neurons feed back, or how do they communicate with the adjacent NPY neurons? And we did an optogenetic experiment to really look at this. And this was, again, going back to what I showed earlier, that channel rhodopsin was injected into POMC-Cre mice. So we have channel rhodopsin being specifically expressed in POMC neurons. And this is the M-cherry labeling. And this mouse is crossed with the GFP-expressing NPY neurons, such that one could shine light and activate these channel rhodopsin fibers and then record postsynaptically in NPY neurons. And these are the sort of experiments, and this is unpublished data that's coming from Todd Stensick, just showing that indeed that when you look at this input and you look at the effects of estrogen, is what estrogen is doing is basically increasing the probability, and in this case, we're giving a single pulse stimulus such that it's releasing glutamate from POMC neurons onto the NPY-AGRP neurons. So this is a classic paradigm that's used electrophysiologically to show that we can increase the probability of glutamate being released with estrogen treatment. And this is now in mice, transgenic mice, but you can see that you have a decreased paired pulse ratio, which equates to the fact that you have more readily releasable pool of glutamate that then can affect the postsynaptic neurons. And this has been looked at now in intact males, ovariectomized females, and you can see that this significant decrease in paired pulse ratio, which is indicative that with this first pulse stimulus, you're getting more glutamatergic release, and as a result, postsynaptic effect on GnRH neurons. So now we're talking about glutamate being released from POMC neurons. So what does that mean, or how does that translate? So Todd's gone on now, and this is very recent data, and it's very preliminary, but he's been able to show that he can treat with STX in the bath, 10 nanomolar STX, and within minutes, this is after 25 minutes, he can increase the probability of glutamate being released from POMC neurons onto NPY neurons. But what's significant is we have an increased probability of release. So what happens now if estrogen's increasing the excitability of POMC neurons? They're firing at a higher rate. So what actually happens under those circumstances? And here's an optogenetic experiment showing that high-frequency stimulation releases beta endorphin from POMC neurons onto the NPY-AGRP neurons. So this is the critical experiment here, showing that now with a barrage of activity coming from POMC neurons, you're actually inhibiting the Arexigenic NPY neurons that will allow estrogen to be truly anorexigenic in the female. And this is shown here that this slow depolarization due to this high frequency 20 hertz, which we published in eLife last year, that this is very effective for releasing peptides from these arcuate neurons. You can see this slow depolarization, and this is inhibited by naloxone. Now this residual amount that we think is due to the metabotropic glutamate effects, and that in NPY neurons, metabotropic glutamate is group 2, so it's inhibitory. So a combination with increased excitation of the POMC neurons, we got beta endorphin release, but also high levels of glutamate such that we're inhibiting the NPY neurons. So this is the pathway that we've worked out in the POMC neurons, this GQ-coupled estrogen receptor that greatly increases the activity of these neurons that results in more neurotransmitter release that have a downstream effect, but also to inhibit the adjacent NPY neurons such that you're inhibiting the anorexigenic driving activity of these cells. And we know optogenetically that if you stimulate the NPY neurons, Sternsen's has shown this, Palmer's group, a whole slew of people that they very robustly increase feeding. So you want to inhibit these neurons if you're truly going to maintain this anorexigenic activity. So what happens then, we measure the effects of this rapid estrogen signaling going north to the membrane, but I have to remind people that we can also get down into the nucleus going south here via multiple pathways. And Olena Runniclave's group showed a number of years ago that, in fact, with estrogen treatment, you have beta endorphin synthesis highly upregulated in these pro-opioid melanocortin neurons after 24 hours. And with this increased excitability, and this is a time-honored model that if you take a female, overreactomize them, we've done this now in mice, guinea pigs, and rhesus macaques, you see this increased food intake, weight gain, etc. If we give estradiol, we can diminish that food intake, that weight gain, and this is just showing one of our first experiments using STX in vivo to like 17-beta estradiol to increase that weight gain. And this is just showing now taking average food intake in guinea pigs. We house the guinea pigs in these tubs because they're gregarious and they're unhappy if they're singly housed, so behaviorally we keep them together and that keeps them happy. And just showing that STX, like estrogen, decreases food intake to maintain weight. So now I want to turn, are there any questions on so far? Okay, so now I want to talk about how this estradiol signaling interfaces with the metabolic hormones insulin and leptin. And I really only have time to talk about or highlight a couple of findings that we published in Cell Metabolism. For a number of years, it was thought that insulin is actually inhibitory to the pro-opioid melanocortin neurons. And it was due to the activation of the KATP channel. But the problem was with those findings with these groups were being translational and therefore using formulations of insulin. And what they didn't account for was the zinc in the human formulations. And we know zinc very powerfully activates KATP channels in neurons. And if one, so we said this didn't make sense in terms of the whole animal physiology because insulin, like leptin, is anorexigenic and therefore should have the same actions on POMC neurons. So we first published the leptin effects in 2011, and then we came back with this cell metabolism paper in which we used purified guinea pig insulin from Al Parlo. And this is what I call the semi-blind patch recording in guinea pig neurons, in which we go in, we target POMC neurons based on their electrophysiological footprint, then we record a response, harvest the cells, and then size them up in terms of how they respond. And invariably, purified guinea pig insulin cause an inward current. That is, starting here, it depolarizes POMC neurons, causes excitation of the cells. And now we've repeated this in the rhesus macaque, doing the same sort of protocol, that insulin depolarizes pro-opioid melanocortin neurons, causes an inward current, and in fact, it activates this family of channels called the transient receptor potential channels. And it shows this weird IV, this double rectifying IV, but it's a cation selective channel that robustly activates POMC neurons. And this is just showing another figure from that paper, but we could block these effects of insulin with a non-selective TRP channel inhibitor. We could activate this channel with rosiglitazone, which is known to interact with these TRPC4 and 5 channels. This is a PI3 kinase. We could block these effects of insulin with the PI3 kinase inhibitor. And this is now doing the biophysical experiment showing this double rectifying IV due to the activation of these TRP channels. But the really critical experiment is that we can potentiate these effects with lanthanum. Now, lanthanum blocks all other TRP channels known to man except TRPC 4 and 5, but it potentiates these effects of TRPC5 in these POMC neurons. And of course, what a lot of people like to see is that our TRPC5 channels co-localize in beta endorphin neurons, and indeed they are. This is just showing with a selective TRPC5 antibody, you can very nicely get show localization. We measure the mRNA levels, but we can also measure the protein levels. And if we look at, and the reviewers had asked for this, so in the same population of POMC neurons, can we see both a leptin and insulin response? And indeed, this is just a current clamp study in which we apply insulin, we get a depolarization, we repolarize the membrane artificially to stop it from firing, and we hit it with leptin and we get further depolarization. So this is the model that we published in Cell Metabolism, that we have both leptin and insulin receptors in POMC neurons that are coupled to the activation of these TRPC5 channels. And recently, the UT Southwestern group has shown that the TRPC5, if they selectively knock out TRPC5 channels in POMC neurons, they did this in male POMC neurons, this causes a decrease in energy expenditure, an increase in food intake and weight gain. So these are just mice that have been on a normal diet. So they've come around to recognizing the fact that these, at least that leptin and serotonin are signaling via this TRPC5 channel. So in this last part of my talk now, I want to get into how does estradiol interface, how does this anorexigenic hormone in females interface with the anorexigenic metabolic hormones, leptin and insulin, specifically in POMC neurons. And everybody knows about the metabolic syndrome and what's at the core. And top on anybody's list is this hyperglycemia and insulin resistance. And this is, you also have the large waist circumference, visceral adiposity, elevated fasting, triglycerides, dyslipidemia, elevated blood pressure, and reduced HDL cholesterol. And this was published in a white paper from the Endocrine Society. But what I had to convince my program at NINDS, and Istan and I were talking about this earlier, was that, that they should be interested also in insulin resistance in the CNS because of the peripheral neuropathy, stroke, et cetera, and all the sequelae that happen as a result of this. And that neurons, similar to fat cells, can show this hyperinsulinemia-induced insulin resistance. And the interesting thing is that males exhibit a higher incidence of metabolic syndrome than females until women go through menopause, and then they catch up to their age-matched male counterparts. And so we started these experiments going back to our favorite model, the guinea pig model, because this is, again, a really nice reproductive model, having this long reproductive cycle. And we looked at the ability of ICV insulin to reduce food intake, the increase energy expenditure, et cetera, using metabolic cages. And these experiments were done with a collaborator, Ed Wagner, at Western Health Sciences Center. And what you see here, if these guinea pigs are put on a high-fat diet, this is 45% fat based on calories, that in the intact males, that if you give ICV insulin, you'll decrease food intake, you'll increase oxygen consumption, CO2 production, the whole 9 yards. I'm just showing you these two parameters for sake of brevity. And you put these male guinea pigs on a high-fat diet, that after 6 weeks you lose this response to insulin. It has no effect. Whereas in the females, now these are intact cycling females, that you do the same scenario after 6 weeks, you see that in the controls, you see a nice decrease in food intake, the increase in metabolism, and also in the high-fat dieted females. So these are now cycling females, and you can see that insulin is still effective. So the question then became, how is this doing this? And so we moved to the transgenic mouse in order to delve into this, and this is just showing the mouse model. These are now transgenic mice, POMC, GFP expressing, such that we can selectively target these mice and do a number of manipulations, but also molecular biology and analysis. And as everyone's published, you put them on the high-fat diet, you see this increase in weight gain, increase in fat deposition. You do a peripheral glucose tolerance test, and indeed, they become glucose intolerant, these high-fat dieted males and females. But in terms of the central effects of insulin, you see a very much different scenario. So if you look at the insulin response, now this is now voltage clamp experiment, so this inward current that's generated by insulin is causing a membrane depolarization and increased excitability of the POMC neurons. And you see that in the males, indeed on a high-fat diet, you lose a central insulin response. So here's in the control diet, this is in voltage clamp, inward current, and now in the high-fat dieted animal, Zippo, no effect. And you can see that you greatly reduce the insulin response. So these POMC neurons in the male have become insulin resistant. That is, they're not responding to insulin. But in the female, what you see is in the control dieted female, you still see this inward current. And in both the control diet and high-fat dieted females. And if you look in current clamp, that is at the excitability of these neurons, and now we're puffing on insulin to actually activate these neurons, you can see this robust excitation in current clamp. So insulin's still very effective in the females to depolarize and excite these neurons. And you can see the graph here comparing these two. So how does then estrogen protect POMC neurons? And this is all, just to remind you, unpublished data, but the clue came actually from a colleague out at the Primate Center, Paul Kivett, from a publication that back in 2006 where there's enhanced leptin sensitivity and improved glucose homeostasis in mice that have SOX3 specifically knocked out in POMC expressing neurons. So SOX is the suppressor of cytokine signaling 3. And we know with fat deposit, increased leptin secreted from fat cells. Leptin gets into the hypothalamus very readily. This is in the leaky blood-brain barrier area of the brain. It causes an upregulation of SOX3 in POMC neurons, and this really feeds back and inhibits this signaling, JAK-STAT signaling pathway that was very nicely shown in terms of the binding to the leptin receptor in this Nature Structural Biology paper. So the first thing we then asked was, is estrogen preventing this upregulation of SOX3? So we moved to the single-cell RT-PCR, and this is now quantitative qPCR done by Olena Runniclave's group, where they harvest 5 POMC neurons, and then they pool the cells, and then they do quantitative PCR to look at the expression of the different neurotransmitters. And lo and behold, what they found is that indeed, that in the male you have, on a high-fat diet, you have an increased expression of SOX3 mRNA, and whereas in the female there's no increase with a high-fat diet. And this is just measuring another transcript, the P110 beta that's involved in the signaling pathway. And the other thing that they were able to pick up, and that's critical for this whole TRPC5 signaling pathway, is the fact that a collaborator in terms of the excitability of these neurons, these POMC neurons, is this T-type calcium channel, CAV31 and 32. So this is the low threshold calcium channel that's activated at a more hyperpolarized level of the membrane potential, but it's critically involved in these TRPC5 channel activity. And not only is the mRNA greatly upregulated, but we also see the T-type calcium channel. This is now whole cell recording, measuring this activity in POMC neurons. And this is just showing the difference between CAV31, 32, and 3 in terms of the kinetics. So 1 and 2 are the two that are upregulated, and indeed you can measure this increased activity. So where we're at this moment then is what we know in terms of the pro-opioid melanocortin neurons, what estradiol is doing. And we have this GQ membrane estrogen receptor, but they also, I should say that they highly express ER alpha, the pro-opioid melanocortin neurons. So we know with chronic treatment of estradiol that you see a decreased SOX3 expression, increased CAV3, 3,2 expression, and we know that there's an increased POMC expression, going back to the 1990s. And ultimately then, this is going to increase pro-opioid melanocortin neuron activity that will then very robustly preserve these anorexigenic effects of the pro-opioid melanocortin neurons in the estrogen state. So just in terms of summarizing, it quickly took you through the fact that estrogen and STX will desensitize mu-opiate and GABA B receptor and hypothalamic POMC neurons via this GQ membrane estrogen receptor. But in contrast, this STX, and we don't know the receptor at this point, it sensitizes the GABA B and response in NPY-AGRP neurons, that is, and as a result, decreasing their excitabilities, which is exactly what you want to do in terms of this steroid feedback. So this net effects is that there's an increased activation that leads to a robust decrease in food intake, weight gain, in gonadectomized females. And I should add that we've also published that this works in STX per se, works in gonadectomized males. To limit food intake and weight gain. And then insulin and leptin signal via this PI3 kinase PLC signaling to converge and activation of TRP channels. And as a result, E2 augments insulin secretion or signaling through this upregulating this T-type calcium channel, enhancing TRP channel activity, and therefore protecting against insulin resistance. And lastly, these are the critical members of the lab that were involved in these experiments. Eric is at Colorado State finishing up his veterinary doctor degree. Casey's an assistant professor now at NC State. These are people that are still in the lab. And I want to recognize the fundings from NINDS and the National Institute of NIDDK and collaborators Ed Wagner and his student down at Western Health Science University, and also we collaborate with Richard Palmer's group in terms of these optogenetic experiments. And this is my lifelong collaborator, Alina Runniclave, sitting perched up on a mountaintop above her farm in Norway. This is a fjord. And this is our son, who's not interested in such mundane activities as climbing up a mountaintop, but involved in the first descent of a what looks like Niagara Falls in Norway. He's actually there in his kayak, and this is afterwards. He got a little bump on his head from this descent, from hitting his head on the kayak. So thank you, and I'll take questions. So yes, Nancy.
Beautiful body of work.
Thank you.
I have a question about the study in which you induced insulin resistance in the POMC neurons with a high-fat diet.
Yep.
So if you were to take those animals and put them back on a regular diet, how quickly will those animals shift back to being responsive to insulin? And so the question really gets at, is it a matter of them losing body weight or body fat, or is there a quick signal that's occurring due to the high-fat diet?
Yeah, that's a great question. We haven't done that experiment to take them off the high-fat diet and see if we can get back their response. But what we would, I guess, speculate if we could reduce some of the fat in terms of the adiposity, limit the leptin expression and secretion, that we would reduce the expression of the cytokine. That would greatly help, and then then you would have the rest of the like estradiol or testosterone being aromatized to estradiol, be able to take all over in terms of the going back to not being insulin resistant, and certainly that that my clinical colleagues claim that if you can get, or argue that if you can at least get patients that exhibit these type 2 diabetic insulin resistance, that if you can just get them to lose 10% of their body weight, go on a diet and exercise, etc., that it greatly reverses this insulin resistance. Maybe, Richard, you could comment on that. No.
That sounds very rational.
Yeah, this is from Ellis, by the way. It was Ellis Levin, so it was a...
So just a quick follow-up,
Yeah.
sorry. So patients who are obese that go under the Roux-en-Y,
Yes.
their insulin resistance reverses before they lose significant body weight. So, there's a lot of interest in what are those quick signals.
Right. Yeah, so no one really knows at this point, what are the quote-unquote quick signals. So, whether it's, emanating from the gut, some of the gut peptides, et cetera, feeding back. So, and there's actually a former fellow at Rutgers, Troy Repke, who's very much interested in that question, in terms of the gut peptides and the feedback on this whole system and the interaction with, steroid hormone signaling. So it's really an open question. I think the key is getting NIH funding to
That's the key to everything.
Yeah. Yes.
From your electrophysiology studies, it seemed that STX was more potent than estrogen as an anorexigenic signal, but the animals didn't eat less.
Yeah, so we think that there's a number of factors that terms that go into that. So, and this is something we're looking more carefully at those were group house guinea pigs, and so whether there's some outliers, in that, in terms of, and this is a long story in terms of the pharmacokinetics of STX. So these experiments, we were giving sub-Q injections of STX in a vehicle that we were really forced to, when we went to primate studies, to not do or not be able to do. So we had to move towards oral administration, and we can get the primates to readily orally take in STX. So now we're going back and looking in mice that we can individually house and individually monitor to look at oral administration of STX and actually see, how it compares to 17-beta estradiol. So we don't know, like from those data group house, whether there was an outlier, whether there was an animal not getting the dose of the STX. So we're repeating that in mice because they're a hell of a lot cheaper than guinea pigs. Guinea pigs, I might add, cost us $2.50 a day for housing per guinea pig. So they're very expensive. Yes, Chris?
So the POMC knockout has that phenotype of being a little overweight, right?
Yeah, yeah.
But is that totally recapitulated with a new knockout? I don't think it is, if I remember.
Yeah, I don't know the answer to that. I'm sorry.
I don't think it is.
Yeah,
So
yeah.
I'm wondering whether there's a combination of mu and another receptor, delta maybe, which is
Yeah, so that raises a really interesting point. So what I didn't talk about is the kisspeptin neurons that exist down in the arcuate, and they are also involved in energy control of energy homeostasis. They synapse in both POMC and NPY neurons. They have a differential effect on these two groups. They are anorexigenic just like the POMC neurons. Leptin and insulin have the same exact effect, and we published that in the cell metabolism paper, but people didn't really pay attention to that aspect. But those arcuate kisspeptin neurons respond just like the POMC neurons. So we think that they're really critically involved in integrating energy homeostasis with reproduction. So, and they co-express dynorphin. So we don't. So we know that the kappa receptor has a role in these arcuate kisspeptin neurons, but whether there's also a delta receptor, we don't know at this involvement. So, yeah, it's a good question. So, it's a real rat's nest in terms of this organization, and it's really now with these transgenic mice and the optogenetics, we can really start sorting out these synaptic pathways and this communication between these different cell groups.
maybe the optogenetics will help. I think it's maybe causing more problems.
Yeah. Rich, yeah.
As you know, peripheral neuropathy is a major medical problem in patients with diabetes. Does your work in the central nervous system offer any clues how one might be able to approach peripheral neuropathies? Are any of these drugs potentially helpful?
Yeah, that's a good question, and we're just at the tip of the iceberg with these POMC neurons, and certainly that if, for example, the UT Southwestern group is really, and it's taken them a while to come around to this, but they're really focused now on the TRPC5 channels. But no one's really shown in the periphery in terms of their involvement and role in terms of insulin signaling in the periphery. There's now some literature coming out with cardiac myocytes in terms of this same class of channels. But whether you could selectively target this in these different peripheral neurons, that's anyone's guess. That this old adage that the selectivity of a drug is inversely related to how long it's been on the market. So it's just like with rosiglitazone and the problems with that, in terms of, I think if the drug companies had done the due diligence and realized that it actually interacts with these channels that are also expressed in cardiac myocytes, that probably they would have been more reluctant in terms of pushing it. So, but at this point, it needs to be looked at peripherally as well. So we're just using it as a model centrally, and we don't argue that it's also the be-all, the end-all for the whole nervous system. So, some other, yes?
Thank you.
Yeah.
For your beautiful talk.
Thank you.
You showed that the insulin coupled with TRPC5 channel in POMC neurons. Do you know, is it knowing that the estrogen or STX signaling also coupled with TRPC5? POMC.
So how is it coupled to the TRPC5 channel? Is that what your question is, or?
Yeah, the question is whether estrogen signaling or STX signaling also coupled with TRPC5.
Yeah, so that what I tried to show at the end here is that there's a collaboration, and this is a very huge literature going back into the 80s in terms of these TRPC5 channels, or these TRPC channels, that they can exist as what are called store-operated calcium channels or ligand-operated channels. And the store-operated idea is that when you deplete intracellular calcium stores, that they're responsible for replenishing calcium. And but they collaborate with other calcium channels, and in the periphery there's a class called ORI channels. But centrally, what we found, but others have published in the cerebellum and other central nervous system structures, that voltage-gated calcium channels are collaborating with these TRPC5 channels, or these TRPC channels. In our case, it's TRPC5. So the idea is, with an initial depolarization, that you immediately activate these calcium channels, these very voltage-sensitive calcium channels, and they collaborate with the TRPC5 channels to generate this huge response, this robust activity. So we think that estrogen and STX is upregulating the expression of these calcium channels that then facilitate the TRPC5 channel opening, that facilitate this whole process.
Thank you. That is very interesting, because I'm thinking whether there's link between the estrogenic regulation of temperature, because there's studies showing TRPC channel involved in that.
Yeah, that's a really good question. And so these, and where it's happening is at the arcuate kisspeptin neurons. So it's this idea that, as we published in the Z-Life paper last year, that these neurons synchronize. They talk to each other via NKB, neurokinin B, and they synchronize this whole group of arcuate neurons. And interestingly, in the hypoestrogenic state, like in postmenopausal or overactimized females, you have an upregulation of NKB expression, but also kisspeptin, also dynorphin in these neurons. And it's this increased synchrony of activity in these cells that are actually, and we think under these situations it's more pathological, that it's driving, it projects up to the preoptic area to the temperature-sensitive neurons, in driving their activity that cause these vasomotor responses. And this is why now in phase 2 clinical trials, Malindo, Takeda, a number of drug companies have selective NKB antagonists that are in clinical trials for hot flash treatment, because these drugs can get across the blood-brain barrier into this arcuate nucleus area and inhibit this activity of these neurons. So, but it's still wide open in terms of, some argue that it's actually neurokinin B receptor expression in the preoptic that is involved directly. But one way or the other, it's these arcuate kisspeptin neurons that are driving this system. And this is why these NKB antagonists are so effective, either in this area of the leaky blood-brain barrier or further up in the preoptic area. So that's a good question. Yes?
Just really quick, while we're still on like the internal cellular
Yeah.
processes that you elucidated earlier. So if I'm not mistaken, you had shown that there was some activation of PKC delta
Yeah.
via the estrogenic response. And if I'm not mistaken, that has been linked to traditional classical mechanisms of LTP. So in other cell types, so I'm wondering if you've looked at those if estrogens via this membrane-mediated mechanism can induce a longer-lasting change than what you guys have seen here.
Yep, yep. In fact, the Einstein group has used STX to show that STX just like estradiol to increase LTP. So it's very effective. So, I should add, and I didn't have time to show the evidence, but the PKC delta will phosphorylate adenyl cyclase 7, and once that happens, it's basically immune from any GI-GO coupled receptors. So normally, if you activate mu, opiate, GABA B, any of these that are, the alpha subunit will inhibit adenyl cyclase activity, but when that's phosphorylated, it becomes immune. So this is why it's so robust and so potent to cause this heterologous desensitization and maintain that activity. So,
Thank you.
all right. Thank you very much.